King Jesus, King Jesus. Okay, I want to share with you some information that everybody should have learned in school. Really, they should have learned it in grade school. Now, forgive me if you know it and I'm telling you something that you don't know. But I believe that you don't know this because the powers that be don't want you to know how simple it is for them to create new diseases to secretly wage war on your life. Now, this picture is a typical picture of a virus. This is the model picture from which all molecular scientists learn about viruses. It is a diagram, a representative diagram of what is called a T4 phage virus that infects the E. coli cell, Escherichia coli, which infects the gut of mammals and most living animals in nature. This is specific only for a certain type of E. coli. Not all E. coli, just certain types. And you can see, as colored in the picture, there are three main parts to this virus. The attachment site, which is the lower part, the blue and red part, and the capsule and nucleic acid, or DNA, which is in the capsid. You can't really see it colored there, but the three parts are the attachment site, which are blue and red, and the green part, which contains two other parts, that is the capsid that protects the DNA material that is contained within it. So the first two parts that we're going to talk about constitutes really the body of the virus. And this is the capsid that contains the DNA. It's pictured here as green for both parts. Now, the capsid is an envelope that contains the genetic material of the virus. And that constitute the main part of the virus, the capsid and the DNA. Now, the third part of the virus is the actual attachment apparatus. Now, this part is specific for a specific cell that it infects. In other words, this virus is not configured to infect all types of E. coli bacteria. It's only configured to infect one type of E. coli bacteria, and that is specified in the DNA that's inside the capsule of the virus, on the genes of the DNA. The genes are contained on the DNA, and one specific gene will code for the proteins that create the attachment site to the cell that is going to infect. Now, that is very simple. It's the simplest type of living organism that reproduces itself in nature. And that's why it's been chosen as a weapon and manipulated as a weapon. Now, that is the example of a virus. And all viruses will have this very same structure. There's no variation. There's only three functional parts of a virus: the protective coat that protects the DNA itself that contains the genes of the virus, and the attachment apparatus that attaches itself to the cell in order to infect it. And it has to be a specific cell. In this picture, we have a demonstration of a diagrammatic demonstration of the 2D and 3D image of the phage virus. As you can see in the 2D image, the DNA is contained inside the capsid, represented as blue in this picture. And infection apparatus at the lower part has a tunnel through which the DNA gets injected into the cell once it infects the cell, once the apparatus attaches itself to the cell. The 3D image, you can see now that the capsid is basically a protective coat around the DNA. So that's a 3D representation of the T4 phage virus that infects E. coli cells, bacterial cells. Not animal cells, it infects bacterial cells. But the same holds true for viruses that infects animal cells, including human cells. Okay, what's depicted here is the life cycle of a virus infection with T4 phage virus on an E. coli cell. You see here that it's a 5-step process. In the first case, the virus attaches itself to the cell specifically. It has to be the right cell that the virus infects. It can't be any cell. It has to be the right one specified by the DNA. It can't attack any other cell, but that type of E. coli cell that's specified by the DNA that's inside the capsule. Once it attaches, the virus can then inject the cell with its DNA through the tubular structure that's part of its infection apparatus, a part of its attachment apparatus. Through that, it injects its DNA into the cell. Once the DNA of the virus gains access to the host cellular apparatus, it can then create new constituents of itself. It creates the proteins, the structural proteins, the enzymes, and new DNA in order which to assemble new viruses from. So it makes the parts that it needs to make new viruses. Then, in the next step, the virus assembles its constituent parts into new viruses. So it makes new copies of itself by assembling itself, just like the original virus that infected the cell. This sometimes is a self-assembling process, and sometimes it's catalyzed or by enzymes. Once it's made enough copies of itself, it bursts out of its host bacterium, thereby killing the host bacterium, and then each new virus particle can infect a new cell in that manner. So this is the life cycle of a virus infection. It's the life cycle of a virus infection. It's a circular process that will continue in most cases, unless sometimes the DNA will go into your cell and stay there and wait when it has the greatest opportunity to successfully make new copies of itself. It can linger in your body, as the AIDS virus has demonstrated. Okay, this is an early model in DNA phage research where they looked at what actually infects a cell. So they took two models, labeled one on the protein coat and the other they labeled the DNA with phosphorus that they could detect, and proceeded with an infection. Here's the depiction of the label. You can see on the left hand side that the label is all in the protein coat on outside of the DNA, and on the right, only the DNA is labeled. Now, in the second step, you can see that the DNA is injected. On the left, there's no label being injected, and on the right, there is the label being injected with the DNA. In the next steps, you can see that the protein coat fell away on the left, leaving no label with the infection, but on the right, you have the label still residual with the infection. So that is how viruses infect cells, and it's specific for each virus to each cell. And this concludes part 1 of my presentation.